Happy New Year, everyone. So I see that everything seems to be working again. So I want to not only wish you a happy new year, but also welcome to another Saturday live stream. I hope that everything works fine as always. Yeah, today's live stream, what I'm going to do, I'll tell you, I'll be honest, I've got a mix of totally random things that I put together on a little cheat sheet of mine here that I've got here. Yeah, so we're going to see how far we're going to get. Some of you already posted a couple of requests in the chat. Thank you very much. I also I'm going to try to show you a few of these things with more or less success. Yeah, it's a little bit improvised every live stream, so sometimes we're not able to see super exciting things. Sometimes we are. So we're gonna have a look. And because it's New Year, I just want to show you not again crystals. You're gonna say, yeah. I want to show you again a couple of crystals because somebody also requested those. And those crystals that you see now right over here, I just want to show you how I made those because they are a little bit different. Are a little bit different than what you normally make. So these are essentially very easy to make. You have to be a little bit lucky to see them that nicely. Let me go down a little bit with the magnification here, because in a different place, the crystals, they don't look as nice, right? Over here, for example, yeah, it looks kind of messy, right? But over here, just here on the side, I think they look kind of okay-ish. And the question is, of course, now, what are they? Let me go up a little bit with the magnification. I even got different shapes in the past, so you have to try a little bit. Let me change this around a little bit. And I'm going to show you now how I made those because, yeah, it's a little bit different than normal. And what I have done is the following, is I've taken and I'm not going to demonstrate this now, okay? But I'm just going to show you the principle here, okay? Desk view over here. And what I've done is, yeah, you know this, your matches, right? And if I placed some matches over here on the slide like this, and then I lit them, and basically as it burned the red phosphorus, then I quickly removed them, and basically the vapors of the red phosphorus, they started to condense on the cold microscope slide, and they made those crystals. Okay, so you call this this process is called sublimation. You're actually going from a vapor directly into a solid, which are the crystals. So I lighted those matches directly on the microscope on a cold glass slide, and this caused basically those crystals to form. Okay, very easy to do. You only have to, and it's kind of a little bit the drawback, you do have to be a little bit, how do you say, patient in finding actually a nice area. So because those tiny crystals here, because a different area, it looks kind of messy, yeah, like this. Yeah, so you have to go somewhere to maybe you have to search a little bit around, and then maybe you're able to find something, more interesting, like over here on the side. Yeah. Let's go up again with the magnification. You can find again a couple of crystals. Now I'm using DIC, but I'm quite sure that with cross polarizing filters you're also able to get those results. Yeah. Over here. I mean, even in the past, I even got different shapes. Yeah. Oh, okay. Look over here. I have not seen those yet here. They did look actually quite almost a little bit like fragments of a snowflake. Yeah. So are these phosphorus? Yeah, must be. I have to admit, I don't know. Maybe there's also some sulfur inside the matches, right? But it is essentially the material that's vaporized as it kind of deposits directly on the cold microscope slide. So it must be phosphorus. I don't know exactly what combination of chemicals those matches actually have. But it's a pretty, I would say, almost a pretty foolproof and a very quick way of just trying out a little bit of these things, right? Unfortunately, they are kind of small. I'm now using my 60 times objective, so you might not be able to see them very well with a stereo microscope. Yeah. So just saying. Also show the main objectives you use. You can actually see the up there. You can see the objectives that I'm using. In case you don't know it yet, I can quickly run you through. I've got 60, 40, 20, 10, 4, and a phase contrast. I didn't try phase contrast yet. let's try this. I've not tried phase contrast. Oh, okay. Okay, the crystals in phase contrast. Yeah, they kind of are a little bit very bright. Yeah. So just saying a little bit of a, how do you say, that's not a bright field. Yeah, with so you don't even need polarized, you don't even need polarization filters or anything. Yeah. What happens? I mean, actually, I have a polarization filter right now, so that's now with a polarization filter, right? Yeah, and depending on the setup that you have, yeah, I have to make it a little bit. I have to make it a little bit brighter. I mean, I've got DIC. So it adds a little bit of color. Yeah, just wanted to show, share that with you because it's, I think, also a little bit fun. Okay, yeah. So that's kind of a little bit the warm up. I want to show you a couple of other things here. Between two wings of a bee. Oh, okay. Yeah, I'm just reading every now and then. I'm reading some of the comments. So what I want to do is the following. I just want to show you a couple of videos. Now, a video that I got. Thank you very much. I got this from one of my viewers, and I have to tell you, yeah, spermatozoa. I already talked about those in the previous live stream. And I have to tell you, the video clip that I'm going to show you right now has already been published, also on Reddit. And I want to say thank you very much. And I have to tell you, it's, I think, the most done with with an eye scope, so with a standard microscope. And I have to tell you, the video and the image quality is incredible. So I'm going to show you just this clip here, and then I'm going to show you a couple of other pictures that I got from a few of my viewers. Just a second, I'm just checking a little bit. I've got the sheet here. Okay, so if you want to also have some pictures and you want me to share them on the live stream, please feel free to send them. Okay, and I will basically also show them to you. Yeah. So I'm going to now do the following. I'm going to show you now this video of spermatozoa, sperm cells. Okay, so here they are, made with a standard microscope, and I have to tell you, I love it because I'm going to show this also to my students, because we are doing actually this chapter as well, and my students actually have to know these parts of the sperm cells. So what I'm going to do for 5 minutes, I'm going to give you a biology lesson. Okay, as well, because how do you send lengthy videos to me? You can send me over WeTransfer, which is a free service. You can send them to me, and then I can download them. Yeah. So this is thank you very much. Here they are. If you want to comment on those, please feel free. As I mentioned, I've rarely seen or never seen spermatozoa of such a good quality. Image quality, right? What I'm going to do now is I'm going to give you a short 3-minute biology lesson, okay? So I'm going to pause the video because I do need an arrow because, what we have obviously, here are the sperm cells. I mean, the preparation process was quite lengthy. In the email that I got, I mean, this was temperature control and centrifugation and so on. So then you had to wait a little bit for them to slow down, because otherwise they were moving too quickly. But let's pick out any one of these. Yeah. So the email that I got was really in very much detail. I just want to give you a little bit of a short anatomy. A sperm cell has 3 parts, right? Over here, the head part. Yeah. Then there is a midsection over here, and then there is, of course, the long tail. Okay. The head part contains, of course, the DNA. There is a nucleus in here, but the tip of the head is called the acrosome and contains an enzyme. It's important for the actual fertilization process. So I'm fascinated that I'm able, with just a standard light microscope, or over here maybe, that we're able to see those things here. So that's the nucleus is in here. Then there's a cap here with an enzyme, in here called the acrosome. And then the midsection over here that you see, it's a little bit enlarged. It's a little bit thicker over here. This is where you have a mitochondrion to provide the energy. Yeah. So there's a spiral-shaped mitochondrion in the midsection. So you see over here, right next to the head, it's a little bit thicker. Also over here, here as well. Yeah, over here you can also see it's a little bit thicker. And then of course you have the long tail. And the long tail contains protein fibers that are sliding past each other. I'm just going to give you a demonstration using my hands. Yeah, if I'm trying to slide my hands back and forth, like, yeah, again, if I'm trying to, yeah, then you see that, yeah, I'm pressing them together and I'm trying to move them back and forth. And then you see that the hand is moving left and right. Right, and that's basically also what happens with those protein fibers that you have in the tail. They're sliding past each other, yeah, and in this way, the tails are making this movement. Really nice. Yeah, let's continue to watch this a little bit because I think it's really really cool. Yeah, yeah, and yeah, if you just yeah watch this carefully. Some of them actually do seem to be a little bit strange over here, like the one here. Yeah. Okay, now the video is looping again. Yeah. It's very, very nice. Yeah. Ah, okay. Thank you very much. You did not centrifuge them. I just used the centrifuge tube as the sample collection container. Okay. Okay. You can send me an email. It's editor@microbehunter.com, or you can also send me over WeTransfer. But honestly, really, when I first saw this, I thought, wow, it's phase contrast. No, it's not. It seems to be a regular bright field, but really, really good quality. Yeah. So I'm actually quite surprised, positively surprised. We're with standard equipment that you're able to do that. Yeah. So every now and then I'm going to have a look. Yeah. So I want to show you also a couple of pictures that I got sent, and yeah, so and I want to show them to you because it's also using fluorescent microscopy. Thank you very much as well. I got an email also with some pictures attached, which I also want to share with you. So that is essentially it's a commercially prepared slide. So I don't know if you happen to be online as well. And if you're also part of the chat, then you please feel free, of course, to comment. And essentially, this one is the regular bright field. But look at the next ones, because the next one we're using actually fluorescence. Yeah, and look how nice it actually starts to light up. Okay, and another one over here. Yeah, using different techniques, and this one I like a lot. Yeah. So essentially, I have over here. It's actually it's pretty fancy and advanced. So there was an excitation using 340 to 380 nanometers, and then the signals were then recorded at a longer wavelength. Yeah. And then essentially there was also the pictures were also sometimes using a cooling element to reduce the noise, really advanced stuff. Okay, and then using artificial coloring. So that's this. Yeah, and so on. So it's really nice. I did not know this myself that. Essentially, just evidently without using any fluorescent staining, that you're able to get those nice, beautiful pictures here. Okay. So it's a plant cross section. You know what I'm going to do? Just because they're in the corner over here, I'm going to simply make this a little bit larger now. Let's read a little bit what we've got here. I mean, it's the text is over here. So yeah, okay, here it is. It's a Leica DMIRB. Okay, it's an unknown dye because the commercial one. Yeah, and field of view. Excitation, yeah, so you've got all of the wavelengths over here, yeah, so really nice. And look at this, even when you zoom in, the clarity is really nice, yeah. So thank you very much for actually sharing those pictures. Okay, I kind of really. So what are you looking at? This here is the cross section of a plant, it's using ultraviolet light, and it's a fluorescent microscopy. Okay, and you can see essentially all of those parts that you see here are the cell walls that are starting to light up. Yeah. So it's a really nice specimen as well. Okay. Thank you very much to Ever Smaller, who's now basically explaining a little bit on how the video with a spermatozoa was done. Okay. But one of the things, maybe if I can ask you a little bit, some technical questions here, is you mixed the enzymes that break down the proteins evenly. So can you please clarify this? Did you add some enzymes, or what did you actually do here? So this would be kind of interesting a little bit. I also read in your email that the enzymes were involved here to break down the proteins, but maybe you can explain me a little bit more. Okay, because would be interesting. So, did you add some enzymes or so to kind of break down protein clusters so that essentially the spermatozoa are able to move more freely, or what did you do exactly? The enzyme part is the thing that would interest me here as well. Yeah. So that is basically, thank you again for the pictures and for the videos. I want to now move on a little bit, and I'm going to show you now another video that I took just a couple of hours ago, 6 hours ago maybe, because there has been a request for me to. Okay, I'm now. Okay, I'm reading comments now. I see. Okay, I got it. Okay, so the enzymes were present in the semen naturally. I'd have to do a little bit of research on this, because normally what happens sometimes is that the proteins and the spermatozoa start to coagulate when they're out of the body, when they die off. Okay, so that's yeah. In any case, I'm going to show you now another video. Okay, because there has been the request to collect again a water sample from a winter water sample, which I tried to do. Yeah, so this is just basically a little bit of a beautiful winter landscape. And I did find a sample. It's not the most exciting one. Okay, yeah, I think that's supposed to be me taking a little bit of selfie. Okay. Okay, so and it was okay. I mean, over here we had a little bit of snow here in Austria, where I am right now. Okay, it wasn't quite as cold as it looks. But I was kind of a little bit frustrated. Where am I going to find a sample? And then I found this thing here for animals. Right, it was frozen. But I did find some green stuff growing on the side here, and I said, okay, this looks very interesting. And it was actually quite wet because there was still a little bit of liquid water there, and that's what I collected. And here it is. Okay, so I thought I'm just going to put this under the microscope. I have to admit, it's a little bit disappointing. Okay. Not quite as much movement as I kind of hoped. I did check, so I added a little bit of snow, which of course melted right now. But I said, we're going to try it in any case. Yeah, so now the video is kind of looping. Okay. And so here we are. Let's put the matches away. And we're just going to make we're going to take some of this stuff out here, and then we're going to hope for the best. I did find a couple of fungi, a couple of cyanobacteria, and so on. So there is a little bit of stuff in here, but again, not quite as much as I hoped. Okay. Here's my 3D printed stand. And and there are different ways on how you can prepare the sample. Of course, you can use a pipette. But I'm also going to use a tweezer, if I can only get it out of the box. Here we go. Okay. Again, it's not the most exciting sample, but usually I always recommend that you take some solid material and you do this. Yeah. Then hope that something comes off. The rest goes back. And of course, this is maybe let's make this a little bit larger here. Of course, this is way too thick, for making a microscope slide. So you want to remove everything here again. put it back, and the cover glass goes on top. That's pretty much it. It's you see all of those grains here still might be a little bit too thick, but you know what? Who cares? I'm just gonna press it flat. Definitely not good enough. So in this case, I will add. In this case, I will add some. It doesn't work. Why does it not work? Could it be maybe that the tip is kind of clogged? This could be. Yep, this now works. So let's go all the way in from the bottom and then hope that we're able to find something more or less interesting. Again, this is a little bit directly from this little water fountain there for the animals. And usually what happens is if you leave it at room temperature maybe for a couple of days, then whatever microorganisms might be in there, ciliates that move around, they will also start to multiply. But we're just going to have a look here. Okay. And we're just going to hope for the best. Oh, that's the wrong microscope. Here we go. That's the one. We all start at the lowest magnification. Oh, that's the wrong. We have to go all the way down. And as expected, it looks like a mess. Yeah. And no movement, at least not at this magnification. And indeed, I did not find any, moving ciliates, but I did find when you zoom in, occasionally you are able to identify a couple of things in here, but it's by far not the most exciting specimen. Yeah. So the further that we move in, here I can show you a couple of things that I was able to identify. Yeah. Usually those things here, they look very much like so-called conidia, which are the basically the spores of a fungus. Those things here, right? Yeah, yeah. So yeah. So yes, yeah, exactly. Also, those over here. Yeah, if you just look over here, those crescent-shaped structures here also look very much like so-called conidia. Yeah. Yeah, and this one here, very typical as well. Yeah, you can see the individual cells here. So what I'm going to probably, you see, the problem is if I go up now with the magnification yet further, I can't because the cover glass is kind of too high. Yeah. Yeah. So, but you see a little bit, at least at this magnification. Again, lots of conidia and fungi, but honestly, surprisingly few ciliates, flagellates, and so on. I would have expected some rotifers maybe, Okay. Yeah. There might be amoeba there. I was kind of hoping that's possible, but they kind of move very slowly. So, yeah. So this was actually a little bit of a also of a surprise because I did expect a little bit more. Yeah. Let me go a little bit like this, yeah. But that's also something that basically where we learn, yeah. And this here, for example, the fuzzy thing, this large structure here, this looks very much like probably cyanobacteria, yeah, which are kind of growing on the surface, yeah. So, yeah, over here as well. Yeah, complex, like almost like in a biofilm. Yeah, but at least this is also something. Those filaments that you see most likely cyanobacteria as well, right? But at least even if there is nothing super exciting and it looks kind of messy a little bit, still, I think that I learned something. Yeah, because samples can be extremely different from each other. Yeah, just wanted to show that to you as well, that essentially, I was hoping for a little bit more to show you. Okay. Yeah. So I'm going to have a look here. Yeah, if there are any comments that you want to write for me, then please include the statement at Microbehunter, so it's easier for me to find you. Okay, to find your comments. So that is basically the one from today. So what is this green stuff? The green stuff that I collected is basically a combination of all sorts of things, right? It's not lichen. It's a combination of cyanobacteria and fungi and bacteria and so on. So it's some kind of an aggregate, yeah, of what we see here. So it is not a single thing. I mean, of course, there were lots of these things in here which look like photosynthetic cyanobacteria. That's why it was kind of greenish. But there was lots of other stuff in there as well. Yeah. So, but I would say let's move on a little bit because I do also have green fluff. Yeah, exactly. Yeah, green fluff. Yeah. So this is a little bit the how do you say the disappointing? No, actually, I don't know. I shouldn't be saying disappointing. I mean that's just the way that nature is. But sometimes we're all super excited. We want to find worms and ciliates and tardigrades and all sorts of things. Yeah. And usually these are the things that I kind of show also in my my videos. But nature is not always that. Yeah. Sometimes it looks like this. Let me do another one. There has been a request. Yeah. There has been a request also, and I also have a little bit limited, how do you say, hopes for that. But over the past couple of months, I've received a lot of requests. Can you do some soil microscopy? Okay. And I was always a little bit reluctant to do that because a couple of years ago, a company who was doing composting sent me a whole box of different types of compost of soil. And they requested that I make some videos and pictures for them of the different soil types because they needed that for their web page. So yeah, something to show to the people. Yeah, and I got a nice box of different soil types, really interesting stuff. I put it under the microscope, but there was nothing exciting there, really. Of course, bacteria you can always find, but sometimes it can be very difficult to identify if there's so many other soil particles there. So I just wanted to say that the expectations, we have to tune them down a little bit. But in any case, I'm going to show them to you still, and I'm going to now use a different pipette. A larger one, or maybe even a yet larger one over here, one where I cut away the front part, and I'm going to take now a soil sample here. So this soil is commercial gardening soil, and it's from a bag, yeah. So it was moist still, so I've taken it from a moist bag, so it did not dry out completely, and then. About half an hour ago or so, I added a little bit of water to leach out whatever is in there. And so it's also easier for me to pick up a sample. Okay, and I'm going to just do it like this. And this is, of course, way too much. Yeah, so look at this mess. Didn't think of that either. So I'm going to put this, I'm going to rinse this later on, so I'm going to put it away somewhere. And then... this is going to be way too dark, right? I'm not able to see a lot, so I need to get a cover glass. And we're going to do the following. I'm going to now push all of the soil to the side and do something like this. Okay? And then you see that there is a little bit of liquid here. I have to be super careful with that soil. Because otherwise the objective is going to touch that. So I'm going to do the following. Now I'm just going to maybe move the cover glass a little bit over so that it's able to collect a little bit more water. Okay, here we go. Okay, and I'm just going to remove now the soil. I'm making a mess here because So look what I'm doing right now. I'm just going to go like this and I'm just gonna remove it. Here it is. Okay, and Yeah. There's a little bit of water here on the surface as well of the cover glass. I want to get that away as well. So let's have a look at this here, okay? And yeah, again, I did not check this specific sample, okay? But again, because yeah, this has been a request, and let's have a look. Okay, again, I do not think that we're able to see a lot of movement here. But in some of the samples, soil samples, I've actually seen so-called the shells, so-called the tests of some amoeba. Yeah. So let's move up yet further. Okay. And you see that there is still plenty of stuff, yeah, to be seen. Lots of plant fragments, of course. And then, yeah, you can essentially try to guess and figure out what the different parts could actually be that you find. You know what, I'm going to go up yet further with the magnification. Very nicely visible also. You see the jittering motion, right? That's, of course, a so-called Brownian motion, which is the random movement of particles. Yeah. So that is essentially soil under the microscope, right? Yeah. So what I would have to do, and this is actually something that I have done when the company sent me the soil, I actually made a so-called an enrichment culture. Basically the enrichment culture is when I added a little bit of food, and whatever microorganisms were there actually did reproduce. So I could actually see way more amoebae moving around. Yeah. But what we see here essentially are mostly fragments. Maybe there are a whole bunch of living cells in there. Of course, there are, but it can be a little bit difficult to distinguish from other debris. And the reason why I'm showing this is because people have repeatedly asked me, can I not put some soil under the microscope and check if the soil is "quote unquote" healthy, right? But how would you actually be able to determine that? Yeah, so that's a little bit the thing that we have to be aware of, that not everything that is visible under the microscope, whether the soil is healthy or not, how would you be able to see that? Yeah. So, but what I could do is the following. I could switch over to phase contrast again. Yeah. And and then usually what happens is. Not what happens, but sometimes, but again, here it's difficult to see, but bacteria are usually, I'm not saying every one of them is a bacterium, but usually are darker. For example, this one here is, could be some kind of a rod-shaped bacterium. The bacteria appear darker, on a brighter background. Yeah, and with phase contrast, you're able to see that better. Yeah, but again, over here, lots of debris and so on. So difficult to actually make out the what the different parts are. Yeah, and what do you mean by healthy? That's a good point. Yeah. Essentially, what I would consider, and why we're growing in summertime, and also with my kids, we're growing, of course, plants and vegetables, in the garden. And what we always take care of is that the soil that we are using, commercial garden soil, actually is free of, is properly made. So there are no let's say, metals in it, chemical deposits, and so on, right? Because we want to eat the food afterwards. So for me, that is actually, of soil being healthy, would be free of contaminants, which might harm us when we eat the food, right? But these things you cannot see under a microscope. Yeah. So that is now bright field. Yeah. So this is just So if you want to actually analyze the soil, and there's one thing, and I'm not recommending that because that's also not always healthy. But I have actually done experiments before already where I made this a soil water mixture, and I removed the soil particles, and then I used a spray can. you know what a spray can is, right? These things here, right? I put a soil water mixture in here, and then what I've done is I took some toast bread, not the one that was yeah, and then I sprayed it, and essentially then you could actually see the fungus growing on the bread, right? So that's a little bit the thing, right? So so there are certainly plenty of fungal spores yeah, in here. So yeah, it's simply also something I wanted to show you a little bit here, and yeah, one of the things that I could do, of course, I could try to make an enrichment culture. Yeah. So the next thing that I wanted to show you, and that is yeah, a little bit kind of relates to the thing that I already started a couple of weeks ago, and I don't know, semi-successfully, I have to say. Is this and also always also a little bit we have to be a little bit more careful. Yeah, this is, oh, you don't see it again. Okay, so this is basically a, yeah, so I've taken a water sample. I have a problem focusing, it doesn't matter. I've taken a water sample, and what I've added in here is those white flakes that you see here are actually cereal grains. What you should do is, if you want to grow your own paramecia, first of all, you have to make sure that there is plenty of oxygen available. That's very important. But you have to ideally crush a cereal grain and add it in here. And then, of course, you're growing bacteria, obviously. And then ciliates that are in here will also feed on the bacteria. So what I'm going to do now is I'm going to just see how well they actually have grown. A couple of days ago, there were quite a few of them, but you see there is a natural progression, and sometimes the concentration of those critters changes. So what I carefully do is I go in here, carefully go to one of those cereal grains, and I take up a small sample directly from here, put it on here, and because there are of course bacteria on here, I want to discard the tip. Yeah, and I close this here again so that I don't tip it over. I'll add a cover glass. And again, you've seen this already last week or so, but it's always a good idea to keep on checking every now and then to see if there's some kind of a progression happening. And where is the scope view again? And yeah, let's turn up, because some people want to see some movement. I don't know what this is. Let's add a little color. This is, it's the wrong. Oh my gosh, it's the wrong thing here. So here we go. Let's try to make it a little bluish. I don't know. Is it blue? Blue? Yeah, blue is okay. Yeah. So yeah, what I've see here, that there are now more paramecia, 2 of them, 3. Okay. Which I did not see before as much. And yeah, there are many of them over here as well. And let's go up a little bit with the magnification. Yeah. I don't know what they are, right? These seem to be, I don't know, no, these are not paramecia. Look at the cilia, are much larger. Yeah, in any case, you get the idea. Okay, and now what? Okay, I'm going to show you something. I'm going to show you this here. Hypotrick, yes. Yeah. Let's and sometimes they stop moving, the smaller ones over here, and then all of a sudden, like right now, they stop moving, and then all of a sudden they start they start kind of reactivating each other again. Yeah. So right now they're very calm, and then all of a sudden they Das Leben im Wassertropfen, yes, that's a very nice book. Identification book. And then every now and then, all of a sudden, they start moving again. So they're not dead or anything. No. Let's tap it a little bit. Yeah, see, I tapped the cover glass and whoop, then all of them start moving again. Yeah. I'm gonna do something nasty. Okay. I'm gonna show you the following. I'm gonna show. Okay. I'm preparing a video for my channel, and on this here, these are copper coins that I put into water, because copper is a very strong disinfectant, and microbes don't like it at all. And what I'm going to show you right now is I'm going to show you something nasty. I'm going to show you why copper sulfate is not good for water organisms. I've got some copper sulfate here, and there is usually a warning sign on copper sulfate. Don't dump it down the drain, because it's very bad for water microorganisms, right? And I've got some copper sulfate over here. Yeah. And what I'm going to do is the following. I'm going to see what happens when I add a little bit of copper sulfate to those critters. Okay, and copper sulfate also works at fairly low concentrations. So I'm just going to show you now why they always say don't dump copper sulfate into water, because it's bad for water organisms. Okay, because indeed it inhibits enzymes and whatnot, and water organisms don't like it. Okay, so let's dissolve it a little bit in some tap water. This one goes somewhere else. And let's see what happens. I mean, I already tried this out, obviously. So let's try to dissolve rather, the copper sulfate in a little bit of water. And take a small amount here. And let's go down with the magnification. Over here, let's go down a bit. Yeah, they stopped moving again. So you know what I'm going to do? I'm going to tap them again and see plenty of, Sometimes they even start collecting somewhere on the side. Where are now the larger? Here is one of them, right? So, and what I'm going to do now is the following. I don't know. I don't even know if I'm able to get that to work. I'm going to do the following. I'm going to remove a little bit of the water here from the side first. So, and now I'm going to add a little bit of the copper sulfate. And then let's see what happens. Yeah, see the copper sulfate over here is starting to move inwards and flush inwards. Capillary action. And let's see what happens to those critters over here. As the copper sulfate reaches them. Yeah. Does copper sulfate make nice crystals? Yes, it should. Good point. Let's try it. Yeah. I think a couple of years ago I actually tried to make crystals using those, and then I don't know. You know what's going to happen when the copper sulfate touches those? They start jittering around, and then essentially they die. Yeah, but you can actually see that the action is quite fast. But you know what? Why not try to make some crystals? Yeah, it does take a little bit of time for it to reach. Oh, look at this. Could this be maybe the non-dissolved copper sulfate? Yeah, you see over here the one in the center. Yeah. So evidently somehow the copper sulfate seems to somehow impact the movement. And they start going a little crazy, then they start jittering, and then let's see. They start losing a little bit control, and then, no, you got to be a little bit patient. It's spinning around, spinning around. Still going. It's usually a question of whether the substance actually properly diffuses, not diffuses, but properly spreads beneath the cover glass. Yeah. So let's see what happens. Let's be a little bit patient. So I'll occasionally, while we're look, this one here already seemed to have, oh, it's still a little bit active. Let's go down again. Maybe it's still too far away. A little, okay, let me see. Yeah, it stopped. It stopped spreading. That's why. I'm going to try to do the following. I'm going to tap it again, and I'm going to see. Oh, they still move. Yeah. In any case, I was able to film a couple of those, how the movement changes when they start to contact the copper sulfate. But I think it's here a little bit more of a problem that the substance did not properly spread. So what I might try is I might try to actually remove some of the liquid from the other side. So let's add a little bit more here. And then, you know what? Let's try to make a couple of crystals as well as requested. Yeah, there's a larger one up here. I have another idea. I might not actually add a cover glass. I might actually try to simply observe what happens when I add it directly without a cover glass. Let's try that. Just a second. Here we go again. Let's put this away. I definitely need a new tip. So, no cover glass this time. Let's just put it directly on here. And scope view, here we go. Of course, it's a little bit... Oh, there's lots of stuff here. Let's do it like this. Like this. Okay, so this is now using my 4x objective. Ah, look, there are larger ones here. Many more larger of these. So, okay, I'm going to give it a try now. And let's see what happens. Small drop of this on here. Here we go. So over here, you don't see any movement anymore. And let's try to follow a little bit what they're doing here, because that's right now right at the edge. Yeah. And sometimes what you're able to see, and this seems to be happening here as well, is they seem to kind of sense a little bit the unfavorable condition. They start to move away from it. But some solid crystals. Okay, let's. Oh, look, those they are starting to move a little bit strangely. Okay, let's try this as well. Let's try to add a little bit, a few solid crystals, because you see, it takes some time. Or right on the other side, it takes some time for it to diffuse. Yeah, and here, yeah, everything's dead already. But let's try the following over here, where there's still something alive. I'm going to now carefully open it, and I'm going to try to add now a crystal. If I can get it open. I'm going to try it with a toothpick. Yep, I got some. Oh, I got a lot. So here it is, the crystal. Yep, and if you... Okay, I'm not using a cover glass, so I have to be careful. Here is the crystal. There's something interesting that happens. Do you actually see how the color changes? It's so much browner. It almost looks like there seems to be some kind of a chemical reaction happening here. Yep. It's better to... yeah, you can now see how it's spreading. Yeah, they're just trying to move away from it. Yeah, but they're not... Look, this larger one is already having some problems. Yep. And as soon as they swim into this copper sulfate, which is now spreading from the top, then it's over. Yep, I'm following this larger cell over here. Yep, and it's reversing. This one seems to be a little bit more resilient, and wrong direction, and that's it. Where is it? Yep, I think you get the idea. Yeah. Yeah. So, as you see, as you see, copper is indeed a strong, how do you say, containing a metal is of course a strong disinfectant, and should be careful. But what I would like to do now is because, let me put this away again. Again, this is a request now, and let's try this as well. Why not try to see if it crystallizes? So maybe that's actually one of the nicer things here. So let's put a little bit of the solution on here. But it might take some time for it to actually dry, and for this reason, I'll be back in 10 seconds. Here I am again. You know what this is, right? This is my coffee warmer. And I need to plug it in. So, and then let's try to see how actually this works and whether it makes some nice crystals or not. Yeah, I got this was a quick 10 seconds. Yes, I've got my, how do you say, my cupboard with all of the stuff right next. Yeah. Yeah. In case you're wondering about this coffee warmer, there are many of them available that you use. You can warm using not warm, but you can. The power supply is USB, and I deliberately did not want to get those because this one actually has a electrical power cord connected because it simply is able to heat up much faster, and and because USB is not able to transfer so much power, right? So it's already starting to become hot. You can also it's maybe too hot already. 55, and let's put this one on here, and then we can see how it actually starts to hopefully form, evaporate and form some crystals. And in the meantime, as we're waiting, I'm going to clean up my mess here a little bit, and I'm going to have another look at some of your comments or questions. Okay, so. Does blood of mollusks have copper instead of iron? I don't know, but nothing would surprise me. Maybe somebody can research this. Okay. Okay, here it's. I found it. It's blue in its most common form, copper sulfate pentahydrate. Okay. Oh, yeah, interesting. Now, copper is, of course, also very important for us humans. However, copper, when in the form of ionic copper in water, it actually does harm microorganisms, and that's why I'm actually working on a video right now, and that's why I'm doing this experiment with the copper coins, because I kind of hope that some copper starts to oxidize here. And I wanted to test a little bit the effect of this copper water on microorganisms as well. Yeah. So this is something that I would like to experiment around with, because what I've seen already, where people have thrown those copper coins into a fountain, into a water fountain, that algae around the copper coin did not grow. Okay, because evidently copper ions were given off into the water, and this inhibited the growth of algae. Yeah. Are copper salts also destructive to bacteria? Wouldn't surprise me. However, one has to be a little bit careful, because there are so many bacteria out there that it would probably be difficult to or problematic to generalize, right? Generally, copper, because it produces also so-called oxygen radicals, which are highly reactive oxygen molecules. Therefore, they react with pretty much everything that's organic. Therefore, it's understandably it should also impact on bacteria. Yeah. But I would not know which specific type of bacteria. Okay. So I'm going to put this already under the microscope because we've seen that it started to crystallize here on the, interesting. Yeah, so basically mollusks essentially have copper-based blood. So it started to crystallize over here already. So what I'm going to do now is I'm going to try to have a look at this in the microscope again. And let's see how well this actually starts to make nice crystals. I always have to refocus a little bit. I think here in the corner we already have. Okay. So here it's still liquid. And I'm going to turn off the arrow. Okay. Yeah, I've just checked USB coffee warmers online. Yeah, most of them are USB powered. Nothing against that, but again, I don't know if they're able to provide the same amount of power as if you actually connect it directly to power socket. Yeah. It doesn't kill microbes by breaking cells open. Copper works in different ways. Copper can actually destroy the cell membrane. It can also impact and block certain enzymes. And it also creates those highly reactive oxygen radicals, which are able to destroy also a variety of organic substances in the cell. Okay, so this is, yeah. So yeah, so you see over here, slowly starting to crystallize. Let's have a look here. Yeah, you know what I'm gonna do is I might actually put it back again on the coffee warmer. Excuse me. Yeah. This one actually does become pretty hot. It's yeah, and it's only 55. I don't know to what extent it's actually accurate, because it does feel way hotter than 55. Okay, you can see that. So let's try it again. Again, the wrong. Need to cool this light a little bit. Yep, here we go. Yeah. Again, you have to be a little bit patient. Ah, here we go. Now it's starting to grow. It's starting to grow now. Okay. Nice. Yeah. So maybe we have now basically reached a saturation point. And as more and more water evaporates, the crystals grow. So, okay. So that's also answered now. Yeah. Modern arts a little bit. Okay. I get it. I'm not sure, but I think that 55 might be the temperature it aims to get a regular mug of coffee. That's an interesting point. You know what I'm going to try to do? I also didn't try that. I mean, I do have thermometers, so what I can do is I can actually put a cup of water on here and then check the actual temperature that it gets. Okay. Now see, now the crystallization started to slow down a little bit. Okay, you want to, yeah, yes, there has been a request for a screen. I mean, I don't know if it's OLED or not, but let me do the following. I can actually show you, I will do the following because this has been a repeated request. Let me quickly grab my mobile phone. So, okay, here is my mobile phone. I don't know which display it is, I have to be honest with you. But I'm gonna try the following. I'm going to now try to go to the stereo view. And I'm going to try to find it. Ah, yeah, okay, here we go. Yeah. Now, I don't know which display it is, but I've already put different displays, yeah, basically under my microscope, and you can actually see the different, yeah. You can actually see the different, how do you say, orientation and the different shapes of the pixels. Okay, so let's do the following. Okay, compound microscope, fine. We can try that. Just a second. I hope that I'm able to fit it in here. I don't know if this is not a little too dark. Yep, I think this is indeed a little bit too dark, unfortunately. Yep. Yeah, I won't be able to see it. So I'd have to try experimenting around a little bit more, maybe until I'm able to get it also to work under the compound microscope. Yeah. 60 times. Oh, then I'm worried and it's even going to be even darker. Yeah. So I think I need a white screen or something like that. Let me see. Let me see if I can. Still here. It's a little bit too dark. Okay, so I'll try to get this to work out, and then maybe I can show you next in an upcoming video the different types of LCD screens. So let's do the following here again. Look, this actually looks also kind of pretty. So, yep, that's again some of the copper that says crystallized now. Looks nice. Okay. Yep. Okay. This is actually also quite pretty. But what I do want to do is the following. I do want to try how it actually looks under regular cross polarizing filters. I need to remove the tape here from last week because I had this. So I'm going to now put the slide on my stereo microscope again. So here are the crystals. And now I'm going to add the second filter on top. And I'm just going to drop it directly on top of it, even if I'm now contacting the... And that's how it looks like. So this is just using standard cross-polarizing filters. Let me turn this. See, it's still not quite solid. And yeah, I need to then wipe the filters. See, it's still liquid a little bit. Yeah. But as you can see, looks quite nice. So plenty of things to experiment. Plenty of things to experiment around with. Okay. So what I'm going to do now again is I'm going to... just a second. That's something I could try. Just a second. That's actually a good point. Just a second. I'm going to remove the top polarizer over here. That's actually an idea. Let me check. Just go to a news site or so. I don't know. Let me go to my web page. So I'm going to try it now the following. I'm going to try it again. Here's some white stuff on here. Where is the scope view? So I've now removed the, actually I've now removed the polarizer. And it's still too dark, unfortunately. Huh. Let me look directly through it. Yes. Ha! Okay. Maybe you're able to see it a little bit. Higher magnification is too dark. Yeah, here we go a little bit. Okay. Yeah. So that is actually looking at a white part of the screen. Okay, here we go. Obviously shows that white light is red, green, and blue. Okay. Let's do the following. Let's go again here, and I'm going to put in now the. Yeah, it's too dark. Yeah. Okay. You know what I could try to do? I can actually try to adjust the camera a little bit here. Yep. I'm going up now with the exposure time of the camera. Yeah. Here we go. Okay. And yet higher. Ah, very nice. Yeah, so I really went up with the exposure time of the camera now. Can I focus this even? Yes. Here we go. Okay. Let me see if I'm able to. Huh. Yep. So you can use a color frequency sweep video. Oh, okay. More patterns. This could be. Yeah. The question is now why is it tilted, right? I don't know if this is. And I know that different screens actually have different types of orientations. So I've seen all sorts already, right? Yeah. So. Yeah, so more patterns are, or more array patterns are basically patterns. If you've got a regular pattern and then if you have another regular grid on top of it, then you see, those patterns, and that might be the reason why they are tilted like this, but I don't know. Could it be, yeah? Does a polarizer fit? So the question is now the following. Let's do the following. Let's put this away. I need to now try the following. I'm going to add now a polarizing filter over it. Let me quickly wash this off. So let's see what happens if I add a polarizing filter in between here, and when I turn it. Today we're looking at why honey doesn't go bad and how it remains stable for such a. I accidentally started one of my videos on my mobile. So here we go. I wonder if this actually what happens if I put it directly on here. No, by turning it. Yeah, I don't see a huge difference. It doesn't. It becomes a little bit darker, maybe. Yeah, but not. No, it does polarize definitely. Yeah, but it just makes it darker. So not a very good how do you say effect. Just it does become darker, but then you will not be able to see the pixels at all. Huh? So where's the desk view? Okay, here we are again. So let me see how. Yeah, basically this one is doing, and I think I'm just going to have a last look at this here to see if it's finished crystallizing in the meantime. Yep. Okay. So this is now with polarization. Yeah, I think it kind of more or less finished crystallizing now. Yeah. So just one, yeah. In any case, this is a pretty with a phone that's quite interesting. I mean, in the past, what I've tried as well is I wanted to just for your info, we've got an ebook readers as well, and I tried to obviously looked also at an ebook reader under the microscope. However, I was not able to see those clear patterns or anything. Yeah, so it looked much more diffuse and fuzzy, and I was not able to see very clear pixels all the way. So just basically, telling you that, I was not able to get, a very nice effect, on the screen of an ebook reader. Yeah, so just saying. Yes, there was another. So a little bit now. What time is it? Yeah, there has been at the very beginning. If you scroll up to the chat, there has been a request about microscope objectives. So I thought I'm quickly going to also talk about that, about so-called long working distance objectives. And there was a question of whether I have those. And I thought, well, actually, it's a good point for maybe just 3 minutes or so to quickly talk about those as well. So also because somebody asked about it, what I have over here is the following. It's an Olympus objective, and what it has here is LWD, long working distance. C probably stands for coded, and it's a polarizing, not polarizing, a phase contrast objective. So if you look in here, you see the ring in here, right? So it's a phase contrast objective, but that's not the thing why I want to show it to you. The question is, what does LWD stand for? Long working distance. It means that the distance between the specimen, the slide, and the front part here is pretty large, much larger than other objectives. It's the distance between the objective and the slide. That's called the working distance, and this one is called a long working distance objective. And why do you actually need those? If you look very carefully over here, it says 160. That's the standard, and then it says here 1.2 millimeters. Normally, objectives have 0.17. So if you check your own microscope, you're going to actually find that microscopes, most of them actually have the number 0.17 written on here, and the 0.17, that is the thickness of the cover glass. So you've got the cover glasses over here. So it's 0.17 millimeters thickness. Okay, so no problem with that. But why in the world? What does the 1.2 mean? It's 1.2 millimeters. That's much larger. So when would you ever use that? Okay, and you will use that actually when you have have a. I don't have one here, but over here I'm just going to use this here. If you have an inverted microscope and if you want to look at something, which is directly in a petri dish, right? Then you need a larger working distance, and then the 1.2 millimeter is actually the thickness of the glass here. Well, thank you very much. Okay, again the questions. Yeah. So this is the reason where those long working distance objectives are primarily used are for inverted microscopes when you actually want to, let's say, for example, in cell culture, when you've got your cells growing in a petri dish and you want to actually observe them like this, right? And indeed, it has basically will impact on the resolution. I mean, the numerical aperture is 0.440 because the clarity. And the question is why don't they have long working distance objectives with a higher magnification, let's say with 40 or 60? If you just check in the chat, this was the question. And the reason is because the further away you go from the specimen, the lower the resolution. And with a 40 or 60 times, you have to go really close, right, in order to still see something clearly. And you cannot go that close because of the large working distance, right? So that's a little bit the idea. Is that they are actually very used for special applications, yeah, and generally, the most common one that I've found is a 20 times, yeah. So so used for metallurgical microscopes with epi-illumination, yes, that's another thing. Is when you essentially have light coming from the top. Sometimes you have essentially light being directed directly through the objective itself. This is sometimes a separate area for light being passed on. Or you need a large working distance as well. If you've got light coming from the side, you need to have more clearance. Otherwise, the objective itself is going to cast a shadow, right? So then you also need a bigger distance. Yeah, so just what I want to say is that they have kind of special applications, right? Yeah. So just wanted to briefly mention this because there was a question concerning this. Okay. So let me quickly have a look here again at some of the questions that you might have. You know what I'm going to do is I'm going to put in the scope view again, just that we see some nice crystals. Yeah. I use very deep micro enclosures, so a large working distance would be great. That's interesting. Yeah. Yeah. Okay. Okay. How do you send lengthy? I'm reading some of the questions here. How do you send lengthy videos? You can do that by WeTransfer. A lot of specimens have some small amount of autofluorescence. You just have to expose it for a long time. Okay, thank you very much. What I do have here is the following, and I might want to try this as well. This flashlight here is actually, I don't know if you're able to see that, is actually a UV flashlight. So maybe I'm able, I don't know, also to use that with epi-illumination. I don't know. Yeah. Again, I might have to make myself some kind of a holder here, and then use light from the top, and then actually try indeed with a long exposure time. Yeah. Okay. What would a microscope be used for in soil analysis? I mean, honestly, when I put soil under the microscope before, I mean, some soil was quite interesting, not very interesting, but I mean, I could actually see that some soil samples contain indeed the so-called the shells of certain amoeba, of course, the fungal conidia and so on. You can see all these things. But again, I would say it's interesting to observe them and simply to look at it, what you find. But I don't think that that microscopy alone can really tell you a lot, or microscopy alone can tell you really a lot about the quality of the soil. Because many of the things that actually determine quality, like for example, whether there are certain metals, heavy metals or contaminants in the soil, essentially that's something you cannot see with a microscope. Yeah. And it's actually those chemicals that I'm kind of more worried about, right? So you'd have to do a chemical analysis. Yeah? Have you tried manure compost? Not manure, mature compost. Not homemade one. Okay, I've not put homemade compost, but I did buy. I did use commercially bought compost. Yeah. Probably the more microorganisms, the better the soil. A very simplistic trend, though. That's a difficult question. So the question is soil better if it contains a lot of microorganisms? Well, I would say, I mean, just instinctively, I would say it depends also maybe on the type of microorganisms present, and maybe also on the biodiversity. If there are many microorganisms present, but they're all the same, then there is not a lot of diversity present. Yeah. So I would probably say it's the number of microorganisms is also maybe not really so significant, because if microorganisms are present, and when the conditions are right, then they're going to quickly multiply anyway. right? So they're going to quickly reproduce anyway. So the number of microorganisms will actually change also depending on, I would say, probably on the moisture content of the soil. Yeah. So having said that, there are actually people who are doing the following. There's this called the so-called the tea bag index. I don't know if you've heard about those. The tea bag index is it's actually so a citizen science project where you get yourself some Lipton tea bags, and yeah, they're very standardized, and then you kind of determine and you kind of put them into soil, and then you can actually measure the time it takes for the tea to decompose. So it's a measure of how quick is a certain type of soil able to decompose organic material. And yeah, and then you can weigh the tea bag before and afterwards, and then indeed different types of soil will decompose the tea bag differently. So this is actually an indicator that different types of soils must have different amounts of microorganisms. Otherwise, they would not decompose it differently. Huh? Yeah, copper was used long time ago to coat ships to prevent weeds from forming on the hull. Oh, interesting. I did not know that. Okay. I mean, I've heard, for example, that people in the past, they realized that if you kept water in copper pots, then the water did not, stayed fresh longer. Yeah, because also biofilms did not form as quickly. Okay. Yeah. So yeah, these were just a couple of questions that you've asked, yeah, deep micro enclosures. Interesting. So I can imagine that if as maybe an object is enclosed deep in I don't know, in amber maybe. That would be an interesting way of maybe some insects in amber where you need a larger working distance. I wonder. Yeah. So I'm quickly going through here some of the comments. Okay. Yeah, okay. I think I've otherwise mentioned, I hope I didn't forget any. I think I mentioned more or less all of the comments here. Just another thing that I just want to inform you on. If you visit my main, so-called the main channel, which is the Microbehunter channel. I mean, this one is the Microscopy one. As I mentioned already before, I did some months ago, I did a little bit of a pivot. I now put all of the Microscopy videos on this channel over here. Essentially, the other channel that where I used to do the live streams and put microscopy videos on, I'm now doing microbiology explainers, short ones. So in case you're interested, also check out the other channel. Some of the videos actually did start to take off a little bit. So I will continue to, basically make videos, short explainer videos also on the other channel. And if you have any questions or if you have any suggestions on what you want me to talk about in the other channel as well, then please feel free to mention that. Okay. The next question here. Okay, I keep 10 milliliters of water in a closed plastic case. I see. Okay. Yeah, you have yeah. And how does the junk jar do? The junk jar? Which junk jar? Are you talking about the tiny little, how do you say this thing here? It looks horribly ugly. Yeah. The return of the microscopic mysteries. Yes. This is another thing is I have too many channels and I need to reduce a little bit. But this is the jar that I made a couple of months ago now, where I put some moss, and you can see over here also there is a piece of decomposing wood, and maybe as a last, I think the junk jar is the one we put all. Oh, that's the one that you talk about. Okay. There was actually a fly in here. I'm gonna show you something. Yes, and maybe that's gonna be the last thing that I would like to show you in the next minutes because you were just asking. Several months ago, we dumped all of the remaining jars into a larger jar. And I'm going to show it to you now because it looks pretty bad. Here it is. I kept on adding water every now and then as water evaporated. Not nice, not nice, not nice, not nice, not nice. Look at this stuff here. This is white stuff on here. I think this is, I have no idea what this is. It looks like fungus or something, right? Yeah, so that is basically the jar, yeah, where I can show you, I mean, yeah, algae on the bottom, on the very bottom, some gravel, and then on top of surface, this stuff here. Yeah. I have no idea what it is. I really have no idea. And because I have no idea what it is, as a little as a last little activity today, here it is, a toothpick. Let's try it. I'm just gonna grab some of this stuff here. Wow, this is a lot. I think that these might actually be some kind of, I don't know. It looks like fungus growing on the surface. So of course, I need a little bit of water as well. I have really no idea what it is. So, this is clean. So, and let's give it a try. Need to turn off the coffee warmer. So here we go. And the scope view. Oh, okay. There is some movement going on. Okay, I just, it's not very smooth because of the exposure of my camera. So let's do it like this. Let's turn it down. Let's turn up the light. Oh yeah, there's some movement going on here. I mean, so there is obviously some life in here, but I wonder what those, actually more than the other one. But what is this? You see all this, those big flakes? Yeah? I have no idea. Just looking at it also doesn't quite look like fungus either. I thought it was, or maybe it's so dense of a network. I have no idea. No, look at this. This is such a dense net. Almost like a biofilm growing on the surface. Interesting, but I actually thought a little bit because the white stuff looks a little bit like I don't know, like the spores of a fungus or something. Actually, it does look a little bit like a fungus. Yeah. Okay. Well, yeah. That's, in any case, that's at the lower magnification. No, this is actually quite strange. Yeah, I'd have to maybe separate it a little bit better, but this looks quite like a dense network of something. Yeah. So there is a little bit of an insect discussion going on here. Okay. I'm just reading the comments again. Yeah, okay. What time is it? 1:34. I think I've been talking now over 1.5 hours already. The hooks between two bee wings, apparently they are activated when the wings need to work as one. Interesting. Okay, so the front and the hind wings basically are connected together over those tiny little hooks. Okay. Well, yeah, in any case, this is, as a last little activity, this is the so-called the junk jar. Okay. At least from the surface, you can see that there are indeed some critters moving around. because of obviously at the top there is oxygen present. Yeah. But yeah, the white fluff that I've seen over here, I don't know exactly what it is yet. This white stuff here. Yeah. Yeah. In any case, some kind of a strange, looks almost like a biofilm. Yeah. So, people, I think what I'm gonna do is what I'm gonna do now is I'm just gonna call it a day again. Okay. Yeah. Today's live stream was a little bit random, but I hope that in any case that you found it interesting. I want to thank you again for all of those. First of all, for the nice video about the spermatozoa. Yeah, it still fascinates me. Also, thank you very much for the pictures. No, then not this one over here. For those pictures here using UV light. It's very beautiful. Yeah. And yeah, I wish you all the best, and I basically hope to see you again, soon. Again, hopefully next week. What I'm going to do probably again is I'm going to post again a request for you to mention a couple of things that you want me to put on the microscope. Okay, and if I know a little bit in advance, then I can prepare them, the things. Okay, if it's too short notice, I might not be able to get all of the samples. Oh, thank you very much for the donation. Thank you very much. Okay, yeah, thank you very much. Okay, and I wish you all the best, and yeah, again, happy New Year. Okay, and hope to see you next week again. Bye bye.